A very good morning, my dear Biotonians. This is Dr. Vani, and on behalf of Vedantu Biotonic, I welcome all the students to this super amazing bio channel of Vedantu and to this class, which is the Marathon 5. Good morning, and how are you doing? I know there could be all of types of students. There could be 2020 batch जिनके लिए ये मैराथन हो रही है, या 2021 बैच एंड 2022 बैच, 11th 12th और एग्जाम गोइंग सारे बच्चे यहाँ पहुंच चुके हैं. Good morning, good morning, good morning. Remember that this is your one-stop destination for biology. Anything and everything related to biology will be done in great depth with a lot of understanding only in this particular class. Yes, in this, class, this is not just a channel, but these are the classes that we take. It's just like a super amazing regular class. तो सुबह की जो classes लेते हैं बच्चों हम 8 and 10, वो किस लिए लेते हैं? 2020 के लिए. शाम को 5 बजे class 11th, रात को 9 बजे class 12. And today being a Saturday, we have a big dhamaka. You also have physics and chemistry teachers coming today. So start करें आज का super amazing marathon on biotechnology. Done. And guys, don't forget this. Yes, my Aman gang, my Chunnu Munu gang, all the gangs are here. See, look at this. ये पूरी प्लेलिस्ट है बायोटेक की, मैराथन्स की. बायोटेक इज पार्ट 5. वी हैव टेकन 11 सिलेबस के लिए 2 मैराथन्स. मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस की मैराथन हो चुकी है. बायोलॉजी इन ह्यूमन वेलफेयर की मैराथन हो चुकी है. आज हम कर रहे हैं बायोटेक की मैराथन. आई होप आपने सब देख लिए. कितनी गैंग, कितारू, आदित्य. चलिए तो शुरू करते हैं. आज हम ऑसम अपना बायोटेक का मैराथन स्टार्ट किया जाए. Yes, everybody, full josh, chalo. So let's get going. And guys, amazing. मुझे पता है आप लोगों को doubts होता होगा, in restriction endonuclease, छोटे मोटे cosmet, ORI, phagement वाले doubts. फिर आपको doubts होते होंगे what is transformant, non-transformant, recombinant, non-recombinant. फिर आपको बहुत doubts आते हैं. Yes, in the selectable marker, selectable markers में आज के बाद you will have no doubts because selectable marker के लिए we have amazing video. फिर doubts कई बार आते हैं ELISA test में, right? छोटे मोटे how does it happen, what is the function, PCR, all of this we are going to do in a lot of detail today. आज शाम को आ रहा है PCB और PCMB specially for NEET aspirants. अगर PCB लिया है तो अच्छा है. अगर by chance you've taken PCMB, how do you manage? First, how do you choose? And then how do you manage? Suri sir is coming for a very special lecture for NEET 2020. How do you boost your score in physics? Only 2 months are remaining. And how do you boost your score in these 2 months which are remaining? And we've specially got Love sir. NTA Abhyas Chemistry Mock Test Paper 1. लेकर आ रहे हैं Love sir. Yes, everybody. तो ये है आपका 8 AM, 5 PM, PCB, PCMB, 6:30 PM, 9 PM. Awesome, आज बैक टू बैक अमेजिंग लेक्चर्स होने वाले हैं. बट गाइस, हैव यू लाइक, शेयर, सब्सक्राइब? मैंने कल आपको एक छोटी सी चीज बोली थी कि यार कल सुबह वी मस्ट गेट अप विद दिस ब्यूटीफुल न्यूज ऑफ 280K. आपने इतनी सी मैम की रिक्वेस्ट पूरी नहीं की, बताओ. गुड मॉर्निंग गाइस. सो हैव यू लाइक, शेयर, सब्सक्राइब टू आवर चैनल? रोज आप लोगों को याद दिलाना पड़ता है. 900 बच्चे देखते हैं, 400 लाइक्स आते हैं. बताओ, रोज एक रिमाइंडर डालना पड़ता है. टेलीग्राम ग्रुप ज्वाइन कर लीजिए, लाइक शेयर सब्सक्राइब कर लीजिए. टेलीग्राम पे आपको सारी इंफॉर्मेशन मिलती है. Vedantu_Biotonic टेलीग्राम ग्रुप पे ज्वाइन कर लीजिए. एंड गाइस, we've always produced ranks, we've always produced results. It is one channel which is known for ranks and results, right? So please make sure that you become a part of Vedantu Biotonic. लाइक शेयर सब्सक्राइब जरूर कीजिएगा चैनल को. और चलो इसी के साथ स्टार्ट करते हैं. एक छोटी सी इंफॉर्मेशन है. अगर कोई 11th या 12th वाला बच्चा हमारी क्लास देख रहा है तो हमने अपने पूरे प्लेटफॉर्म को, we made sure our entire platform is free for you at vdnt.in/yt live. We will also have a mentee at the end of the class. वो तो होना ही है. Yes, no, क्लास के एंड में हम एक छोटा सा मेंटी करेंगे. स्टार्ट करें पूरी बायोटेक की यूनिट. रेडी? चलो. So let's get started with the principles of biotechnology. Now I want everybody to open your notes, to open your copies. Is that clear? Yes, make sure everybody opens your notes, your copies, and we start with a process which is called as Principles of Biotechnology. We start now. Jahan bhi aapne notes likhe, I will specifically point on to certain things. Those things are very important. Please make sure that you revise them and go before your examination. Biotechnology is the industrial application of biochemistry. Why have I used the word industrial here? Because if the scale is not big, and that is why everything is done in bioreactors, because I want the scale. I cannot make one katori of curd and say it's biotechnology. The scale, the level, the amount of curd, vaccines which I make have to be very high. So I combine biochemistry, I combine microbiology. This is also very important. Please see the human health and disease strategies wala marathon. I have mentioned in detail about all the microbes which are important. We've done MCQs on it also. There is chemical engineering, there is genetic engineering in order to make the best use of the capabilities of microorganisms. If it is genetic engineering, we also have a committee which is called the Genetic Engineering Approval Committee, through which all the approvals have to be taken before I ever begin with any biotechnological setup, culture, tissues, cells, etc. for the benefit of mankind. Plant breeding, which I did in Marathon 4, please watch it, it is very important. Plant breeding pe questions aate hain, questions do come on auxins and cytokinins, remember this. Tissue culture and micropropagation, genetic engineering and genetic modified crops, BT cotton, BT brinjal, microbial fermentation and plant disease diagnostics. In today's class, I will teach you RNA interference. You will never forget it because there is a video on RNA interference. So this is the basic definition. Let us now move ahead. RNA interference will be done in today's class. It's very important. What are the principles of biotechnology? The most important is I need to identify a DNA with a desirable gene. I want to make insulin. I want the DNA of insulin. Once I make insulin in the laboratory, I will call it humulin. The first people to do it were Banting and Pest to identify it. Frederick Sanger was one person who was also associated with them. So insulin. I want to make glucagon. I need to identify the desirable gene. I need to take it out. How will I cut it so that I can identify and take out the desirable gene? For that, I will have enzymes. Those enzymes are called as molecular scissors or restriction endonuclease. Done. Okay. Step 2. I will introduce the identified DNA into the host. The host most of the times is a plasmid, जो कि extra chromosomal genetic material होता है. It's an extra genetic material. We call it the extra chromosomal genetic material. Yes, तो यहाँ से जीन लेकर मैं किसी और के अंदर उसको add करूँगी. Correct, everybody? Right? I will add it somewhere else. Guys, are you understanding? Give me a high five. Done. 1100 people nearly watching the lecture. Vani ma'am's got 700 likes. How will I know you are enjoying the lecture? Come on. I need this class to have a feedback mechanism. चलिए, ये YouTube lecture नहीं है, ये हमारा V Biotonic का classroom है. We talk to each other. गलती से कोई ब्लॉक होता है उसको अनब्लॉक करते हैं, आपके सवालों का जवाब दिया जाता है. ये हमारा classroom है. YouTube recorded lecture नहीं है. It's live classroom. चलिए, give me a feedback whether you are understanding or not. Come on, that is the way the teacher will also get the energy in the class. चलो. Excellent. आ जाइए. And we maintain the introduced DNA. सबसे पहले यहाँ पे sterility भी होनी चाहिए. Sterile conditions wherever you add and introduced DNA को host में डालेंगे and we will create a recombinant progeny. So I need desirable gene, I need a carrier, and I need to combine both of them in a sterile atmosphere. So what do I need? Desirable gene, I need a carrier, and I need to combine the two in a sterile atmosphere. Cool, everybody? चलिए आगे बढ़ते हैं. Now, what are restriction enzymes? Listen to me very carefully. किसी जमाने में एक बेचारा बैक्टीरिया था. एक बैक्टीरिया था कहीं पे किसी जमाने में. There was one bacteria. उस बैक्टीरिया पे बार बार वायरस का अटैक होता था. So over a period of millions and billions of years, this bacteria, listen to me very carefully, this bacteria to protect itself made an enzyme which used to destroy the genes of the virus. It restricted, इसने जो enzyme बनाई, it restricted the growth of viral genome in bacteria. It used to, जो बैक्टीरिया ने अपनी मदद के लिए बनाया, जो बैक्टीरिया ने बनाया for its own safety, it used to destroy the genome of the invading virus. That is why it used to restrict the growth of the viral genome in the bacteria, and therefore it was known as restriction endonuclease. It used to restrict the viral growth. Who made it? We have not made it. It is a natural enzyme which is made by bacteria against the virus. Humans are super clever. 2 मिनट लगाते हैं किसी को कॉपी करने में, हाँ या ना? Humans are the cleverest species. Within nanoseconds we can copy somebody. Yes or no? अब ये वाली स्माइली बना दो. Yes, we can take nanoseconds to cut, copy, paste. Yes or no? So human beings ने बैक्टीरिया की उस restriction endonuclease को कॉपी करके बायोटेक्नोलॉजी में यूज कर लिया. Humans तो शुरू से यही कर रहे हैं, extremely clever होते हैं. Understood everybody? So we have not made these enzymes. ये बेचारी एक बैक्टीरिया ने बनाई थी, that too against virus. But humans went, took it from the bacteria, copied the enzyme and used it for its own benefit. अपने benefit के लिए use कर लिया. That is the history of restriction endonuclease. It comes, and that is the reason why it is called restriction, because it used to restrict the growth of the virus in the bacteria. Write it down. Done, everybody? Yes, done. Chalo aa jao. So it cuts DNA at specific points, does not cut it at any point, cuts it at very specific points. The specific points are called as palindromes, which is something like mom or Malayalam, M-A-L-A-Y-L-M, M-A-L-A-Y-L-M. Right? So those areas are called palindromes, like wow, like mom, like Malayalam. Understood? Yes? Chaliye. Cleaves vector. Now guys, I just have one request. Please don't keep asking मैंने reply किया, तुम्हें दिखा, तुमने reply किया, मैंने दिखा. आइए क्लास में पढ़िए. So it cuts the vector. Cleaving is to cut the vector plasmid and foreign human DNA. Now the beauty is, what is the really important thing that it does is, I would really like to explain it to you. Now suppose these are the two, this is, let us say, human DNA which needs to be cut, and let us say this is gene of interest, or this is plasmid, both of them. Can you see? So it's going to cut both of them. See, it's cut. Can you see? And see this also, it's also got cut, and it will recombine. Now can it recombine? Can you see? It has recombined. Can you see this? It has recombined because it is cut at the same place. Therefore it has recombined. Did you understand, everybody? So the restriction endonuclease will cut the plasmid and the foreign DNA at the same palindrome, which will create complementary bases. Understood? It will create complementary bases and it's going to join. Cleaving DNA makes DNA fragments ending in short. And see, when you cut it, when you cut this, now look at this. When you cut this, देखिए ऊपर AATT C, नीचे AATTC. So these overlapping hands, ये जो overlapping ends है, आपने ऐसे कट किया, ये जो overlapping ends हैं, जो एक दूसरे से attach हो सकते हैं, ये overlapping ends, इनको हम क्या कहेंगे? We call them the sticky ends. We call them the sticky ends. What do I call them? I call them the sticky ends. And the sticky ends allow insertion of foreign DNA into the vector DNA. Did you understand, everybody? So हो सकता है. Now look at this. Do you see a gray and a green recombined? So this is a foreign DNA and I have recombined it because इसके पास भी AATTC है, यहाँ भी AATT है, and they're going to recombine just like I recombined these two. Just like, see, now they're going to be complementary to each other. They're going to be complementary to each other. They are different, see the color change, but they're going to be complementary to each other, so I can combine it. Double stranded, अगर मैंने ऐसे cut किया होता, now if I cut it like this is very important, these come in examinations. If I cut it like this, then I get blunt ends. It's blunt. But if I cut it like this in the middle, only then, if I cut it like this, I use a ब्लैक कलर में। अगर मैंने ऐसे कट किया, देखो सी बीच से, देन आई गेट स्टिकी एंड्स बिकॉज ओवरलैप है। अगर मैंने ऐसे ही कट कर दिया, then I'm going to get blunt ends. I think it's 100% clear. Yes, विकास को पता तो है, विकास तो हमारा पुराना बच्चा है, that mentee is always at the end. So this is very important. What are the sticky ends and what are the blunt ends? Very good. चलिए, let's move ahead. Done? Now, what are the types of restriction endonuclease? And this has come in exams multiple times. This is AIMS 2015 question also. If I cut the DNA at the ends of the DNA, if the cut which I'm making is at the end, it's called as exo. The word exo comes from exit. The entire unit we will do, and we will take tests on this unit, so you will see how beneficial it is. Exonuclease. If I cut within the DNA inside at specific locations, if I do cut it on the inside at specific locations, I'm going to call it endo. The word en stands for entry, which is inside, entry. You're moving inside. Right, everybody? So when I cut it on the inside, I call it endonuclease. When the DNA is cut at the ends, it's called as the exonuclease. Right? We will also do the history of endo-exo. Don't you worry, everything is done. Right? इसकी cheat sheets आ रही हैं, कल cheat sheet में आपको सारे scientists, सारी history सब कुछ मिल जाएगा. Don't worry. They can bind to specific recognition sequence of the DNA, और दोनों strands को specific points बार-बार बोल रहे हो because the specific points are palindromes. Done, everybody? So first is concept. आप मेरे साथ अभ्यास के पेपर सॉल्व कर रहे हो, आप मेरे साथ एम्स के पेपर सॉल्व कर रहे हो, आप मेरे साथ मंडे को एफएमसी का पेपर सॉल्व करोगे. कॉन्सेप्ट 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 कॉन्सेप्ट, दैट्स द बॉटम लाइन. डन. चलिए आइए. सो एक्सो एंडो इज क्लियर. एक छोटी सी अनाउंसमेंट करनी है ओनली फॉर 2020 बैच माय डियर चिल्ड्रन, जस्ट फॉर टुडे बिकॉज वी गॉट सच फिनोमिनल रिस्पॉन्स that I've extended the V special code. बच्चों 90 plus live sessions आपको मिलेंगे. वानी मैम की 170 recordings, 170 पूरा बॉटनी, पूरा zoology, 20 test assignments, question papers, mock test, recorded sessions right till 26th July आपको मिलने वाली है. सिर्फ और सिर्फ अगर आप कोड लगा लोगे V special vdnt.in/neetcc पे जाकर V special कोड, 1 जून से क्लासेस शुरू हो रही है, लाइव क्लासेस प्लस वानी मैम की सारी जो मेरा बैच है उसकी रिकॉर्डिंग, एक एक चीज की रिकॉर्डिंग. फोटोसिंथेसिस मैंने 3-3, 4-4 क्लासेस में करवाया, रेस्पिरेशन 4-4 क्लासेस में कराई. यू विल गेट ऑल द रिकॉर्डिंग्स. केमिस्ट्री पढ़ाएंगे नेहा मैम, फिजिक्स पढ़ाएंगे गौरव सर, पंकुरी एंड अमृता मैम पढ़ाएंगे आपको बॉटनी एंड ज़ूलॉजी रिस्पेक्टिवली, एंड वानी मैम की सारी रिकॉर्डिंग्स विल बी गिवन टू यू फॉर फ्री विद दिस बैच. Right? आ जाओ. अब मैं आपको पहले
restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences.
Can you see GA?
The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other.
Now the voice is fine?
Single-stranded ends can pair with each other.
The voice is fine?
Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease
I will explain it again. Don't worry.
Restriction endonucleases are fundamental tools in genetic engineering.
ligase will come and join. I will explain the video again. Now I will explain the entire video to you again. Don't worry, I've got 5 videos, specially जो मैं video लेकर आई हूँ आपके लिए of the एंटीबायोटिक सेंसिटिविटी और एलाइजा, वो वीडियो तो आप मिस नहीं कर सकते. आपके डायरेक्ट क्वेश्चंस उस वीडियो से निकलेंगे. नाउ यू विल नॉट हैव एनी मोर बफरिंग. मैं आपको समझा देती हूँ, आप सिर्फ वीडियो देखते चलना. यू विल हैव अ पैलिंड्रोम GAATTC. स्पेशली बच्चों जो लास्ट के 2 वीडियो हैं आपके एंटीबायोटिक सेंसिटिविटी, मार्कर जीन्स के एंड एलाइजा के, उनके बाद कभी कोई प्रॉब्लम नहीं होगी, ना मार्कर जीन्स में ना एलाइजा में. So you have GAATTC. It is going to cut from the inside. Now you see, do you understand what is the sticky ends? देखिए ये AATTC अलग हो गया. These are sticky ends because I've cut them. अब कट देखो, कट आ रहा है ऐसे, ऐसे, vertical, horizontal, vertical. और how will you join them? End में देखना, there are 2 separate colors which are coming and joining. चलिए स्टार्ट करते हैं. इज दैट क्लियर एवरीबडी? एंड बच्चों इन इंट्रॉन्स को स्प्लाइस करो, टेंशन मत लो, फोकस करो. चलो.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences.
Specific nucleotide sequences are getting cleaved.
It is often 4 to 6 nucleotides long.
4 to 6 nucleotide long.
Restriction endonuclease EcoR1 recognizes the sequence.
Remember the site EcoR1. EcoR1 site.
The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The 2 strands of the DNA duplex have the same nucleotide sequence running
same
in
sequence, opposite direction.
Recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the 2 fragments are complementary to each other.
They're complementary to each other.
Ends can pair with each other, sticky ends. Once their ends have paired, the 2 fragments can be joined together
by ligase.
DNA ligase,
yes,
which reforms the phosphodiester bonds of DNA.
Ligase will join the phosphodiester bonds.
This restriction endonuclease is so valuable is that any 2 fragments of DNA produced by the same restriction endonuclease can be joined together.
2 different sources.
Restriction endonucleases are fundamental tools in genetic engineering.
That's it. Guys, you don't even need a voice if you want to study. 2 different sources can be joined by ligase, which rejoins the phosphodiester bond. It's an endonuclease. Is it clear? Clear? चलिए आगे बढ़ते हैं. Guys, don't worry. अगली बार से we'll try and increase the voice also. Cool? Right? अगर आप headphones use करोगे, आपको voice बिल्कुल ठीक मिल जाएगी. आइए अब हम cloning vectors पे चलते हैं, because cloning vectors are most important. The most important are cloning vectors. ठीक है, चलो आ जाओ. Now they are the DNA molecules that can carry the foreign DNA. कुछ तो होना चाहिए जिसके अंदर मैंने foreign DNA segment डालना है. What is foreign DNA segment? It is my gene of interest. जो मुझे चाहिए. It is my gene of interest. What is my gene of interest? Insulin, humulin, glucagon, anything. It will replicate inside the host cells. Host cells के inside it's going to replicate. The most common cloning vector is plasmid. The definition is circular extra chromosomal DNA of bacteria. It already has antibiotic resistance gene. It could even be bacteriophages. So ये रहे आपके nothing but the structure of the plasmid. Let us look at the structure of the plasmid or the cloning vector in detail. सबसे पहले जो कि बहुत important thing है. Now see. You need a site which can be cut by an enzyme. What is the name of the enzyme which is going to cut? The name of the enzyme which is going to cut is called as restriction endonuclease. So can I call this site as a restriction site? Can I call the site as a restriction site? What is the restriction site? Restriction site will have palindrome. Restriction restriction site वो site होगी जिसके पास palindrome है, which I am going to cut with the help of restriction site because the name of the enzyme is restriction endonuclease. I am going to cut it. Cleave means to cut with the help of restriction site. Now it is written that restriction site is within the LAC-Z gene. This is the LAC-Z gene and the palindrome is within the gene. So if I cut it and I insert the foreign DNA, will I make the LAC-Z gene inactive? Say yes ma'am, no ma'am. If I cut it and if I insert the foreign gene, almond gang, tell me, will I make it inactive? And बच्चों, are you liking, sharing, subscribing? मुझे तो सच में नहीं लग रहा है कि आप लोग कोई भी अपने चैनल को बिल्कुल भी, अगर आप मुझे तो I don't feel any love in your eyes. Yes, बिल्कुल भी नहीं. Do you do that honestly? सच्ची मैंने कल आपको बोला था 280, आज सुबह हो गया, देखो 280 नहीं कराया अभी तक आपने. Really वैसे इतनी भी मैम की बात नहीं रख पाए आप. पक्का लाइक शेयर सब्सक्राइब कर रहे हो, especially the new ones. If you don't want to miss anything, this is your channel. Make sure you like, make sure you subscribe, make sure you become a part of Be Biotonic, because all these super amazing lectures only happen on Vedantu's pure bio channel, that is Be Biotonic. चलो आ जाओ, let's move ahead. Right, so please guys, make sure आप like, share, subscribe कर लीजिए, because we also need your support, your love back. चलो, so I'm going to cut the restriction site. The restriction site क्या होती है? Restriction site is the site where the restriction enzymes will act. It is a site for LAC-Z gene. If I cut the restriction site with the help of restriction endonuclease, if I cut it, the LAC-Z gene will become inactive. Yes, if I cut, now can there be another restriction site called ampicillin resistance? Yes ma'am. Can there be one more restriction site, let us say penicillin resistance? Yes ma'am. Can there be another restriction site, tetracycline resistance? All antibiotic sites are restriction sites. What you have to remember is restriction sites के बीच में there is a palindrome. So when you cut the palindrome, the restriction site is becoming inactive. If you understand this, you will be able to solve MCQs. So do you see the restriction site blue color, blue? And do you see the foreign gene inserted between gene of interest? Please see that. And the restriction site, see? So I have cut it. See, the blue gene has been cut. So it means the blue gene has become inactive. And now I will put this in the vector. Recombinant vector carrying my gene of interest. Do you see that? Recombinant vector. अब मैं इसको mix करूँगी. How will I join them? I will obviously join them with the help of ligase. I am going to mix them. I will get a recombinant vector. Now the vector should have something like origin of replication, from where replication starts. The vector should have a high copy number because it should be able to make many copies for me. The vector should also have a restriction site where I can add the foreign gene. So let us look at the properties of the vector. What are the properties of the vector?
Plasmids can be good cloning vectors.
Why plasmids are good cloning vectors?
Replication and are.
What I'm going to do is, I'm going to switch off the volume and I'm going to explain this on my own.
Plasmids can be.
So let's do it on my own. Now plasmids can be very good cloning vectors. Why should plasmids be very good cloning vectors? One second guys. Why are plasmids very good cloning vectors? Because plasmids have the origin of... One second, what's happening? One second guys. Yeah. Doesn't want to... Try it one more time. Okay, let's switch on the volume. It doesn't like switching off the volume. It's got angry, wants to talk to you. So let me do that.
Can be good cloning vectors because they carry an origin of replication and are.
They do have origin of replication.
Independently within a cell. Most plasmids used as vectors also encode some type of selectable marker.
Selectable marker. What is the function of the selectable marker? What I'm going to do is I'm going to stop and explain it again and again. So you see what happens is, we'll try and do something in the next class about it. Don't worry. The moment you stop it, it goes for a run. Yes, it says, see, look at this. Just look at it. जल्दी जल्दी खत्म करो, सैटरडे आ गया, वीकेंड, मुझे छुट्टी चाहिए.
Plasmid.
Right, probably यही चल रहा है, but I will. Yes, everybody, never mind. So plasmids, let me give you the gist of the video and then you will understand it. So all plasmids have a very, very good. Yes, let me make it full screen. मैं एक बार समझा देती हूँ आपको यहाँ पर जाकर, and then we'll come back. कि है क्या? ये भी एक वीडियो है? नहीं, ये स्लाइड है. चलिए, ठीक है. हाँ जी. So मुझे प्लाज्मिड्स के बारे में बताना है आपको. क्या क्या आपने ध्यान देना है? प्लाज्मिड्स, सारे प्लाज्मिड्स के पास ओरिजिन ऑफ रेप्लिकेशन होगा. बिना ORI के रेप्लिकेशन स्टार्ट नहीं हो सकती है ना. सच्ची बेचारा, आई थिंक रियली. अब ये सोचो, रोज 15 क्लासेस नियरली, आई एम नॉट जोकिंग, सुबह 6 o'clock से लेके इवन ऑन अ संडे till 10:30 p.m. every day 6 a.m. So you know, machines also have a life after all. Yes, आ जाइए. नेवर माइंड, मजाक हो गया, थोड़ा सा मजा आ गया, थोड़ा सा क्लास की, you know, you were studying, that little change has happened. So आ जाइए, let's focus back onto the class. So all your plasmids have to have an origin of replication because it starts from here. Without an origin of replication, you cannot start any replication. At the origin of replication, you really require a high copy number. What do you understand by a high copy number? You don't want only 1, 2, 5, 10 copies. You want hundreds and thousands of copies of the same gene of interest. So you have a very high copy number. After that, they do have a selectable marker. They do have something which is called a selectable marker. With the help of the selectable marker, we are able to distinguish between transformants and non-transformants. Very, very, very important. I will tell you all of this in detail. Is that clear? Yes, don't worry about anybody. बच्चों, आप अपनी पढ़ाई पे ध्यान दो. बहुत इंट्रॉन्स आएंगे आपको परेशान करने. तो we have a selectable marker. What do we have? We do have a selectable marker. Selectable marker usually मैं आपको अभी इस beautiful video में दिखाऊंगी. It could be antibiotic resistance or it could be color change gene. Antibiotic resistance is difficult to see because it is not a visual selectable marker. दिखाई नहीं देता है, so it is slightly on the difficult side to see. Whereas the color change is very, very easy to see. Very easy to see the color change one. Done. तो अभी हम इन्हीं चीजों पर on the video we will talk and let's talk in a lot of detail. बिल्कुल पुंगोलू, good. चलिए आइए, तो एकदम से हम वीडियो पर चलते हैं and we will understand it.
Cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell.
Independent replication.
Plasmids used as vectors also encode some type of selectable marker.
Selectable markers.
The gene for resistance to ampicillin.
Ampicillin resistance. Ampicillin is an antibiotic.
The host cells are ampicillin sensitive. The only host cells that can grow on a medium containing ampicillin are those that have taken up the plasmid. Vectors must also have a small sequence of beta.
So what did it say till now was, देखो, the plasmid only will have antibiotic resistance gene. Very important. कभी भी antibiotic resistance की gene बैक्टीरिया के जीनोम में नहीं होती. The antibiotic resistance gene cannot be there in the genome of the bacteria. Where is the antibiotic resistance gene? Come on, tell me. Plasmid. So if a bacteria does not have plasmid, will it have antibiotic resistance? Yes or no on my screen. If a bacteria does not have plasmid, will it be antibiotic resistance? No. If a bacteria does not have plasmid, will it be antibiotic resistance? No. I want to show you this now. See here. See. See. See, it is written here.
Ampicillin sensitive.
Ampicillin sensitive host cell. So what did that ampicillin sensitive host cell meant? It meant that that bacteria cannot It cannot, right? It cannot do any antibiotic resistance. Good. Chaliye.
Plasmids can be good cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell. Most plasmids used as vectors also encode some type of selectable marker, such as the gene for resistance to ampicillin. If the host cells are ampicillin sensitive, the only host cells that can grow
sensitive, it will die in the presence of penicillin.
That have taken up the plasmid. Vectors must also have a small sequence of base pairs that can be recognized by a restriction enzyme.
See?
When this enzyme opens the circular plasmid, foreign DNA
See?
can be incorporated.
Sticky ends. Sticky ends.
The plasmid vector and foreign DNA are both cut with the same
complementary
and mixed together. Not all molecules will
see foreign DNA.
Recombinants. Some vector molecules will reanneal without incorporating foreign DNA.
Had new DNA, no new DNA.
Cells that contain plasmids that have incorporated foreign DNA. A second marker gene is needed on the vector.
I want to stop the slideshow here.
The restriction enzyme site within its nucleotide sequence.
Okay, now see. Did you see? There are two antibiotic resistance genes, or there are two selectable marker genes. Did you see antibiotic resistance, ampicillin resistance gene? Yes. Come on, everybody. Epic video no Pungolu. क्या आपको दो selectable marker नजर आ रहे हैं? एक ampicillin resistance gene जिसको मैंने touch भी नहीं किया. I've not done anything. इसमें भी ampicillin resistance, इसमें भी ampicillin resistance. Both the yellow ones are plasmid. ये रही LAC-Z gene, ये रही LAC-Z gene. How did I cut it? I cut it in the middle of LAC-Z gene. Where did I cut it? Tell me. Amazing video. मैंने कहाँ पर cut लगाया? Where did I cut with the help of restriction endonuclease? In the middle of LAC-Z gene. But here, the foreign gene. See the green colored foreign gene, new DNA. The foreign gene did not enter. Here the foreign gene entered. So because the foreign gene entered, here the LAC-Z gene has become inactive. Yes or no? The LAC-Z gene has become inactive because the foreign gene has entered. That is called as insertional inactivation. You will never forget it now. That is called as insertional inactivation. Is it clear? What is it called? It's called as insertional inactivation. Did you get that point? And here, there is no insertional inactivation. So LAC-Z gene will produce a blue color. So out of these two, this will produce a blue color, but because here the LAC-Z gene is inactive, it cannot produce a blue color. So will I be able to tell the transformants from the non-transformants? Because the non-transformants are going to make blue color for me, whereas the transformants cannot make a blue color because the LAC-Z gene has been split. Say yes or no. Is that clear? Say yes or no on my screen, please. Yes, everybody. Nail the concept. Is this concept nailed? So you have a plasmid. All your plasmids will have antibiotic resistance or the LAC-Z gene. Your restriction endonuclease may cut the restriction endonuclease will cut the genes like LAC-Z or the ampicillin resistance. If the LAC-Z gene are cut and the new DNA is entered, it is called as transformant. LAC-Z gene was cut but the new DNA did not enter, so it is called as non-transformant. Here the LAC-Z gene is functional, but here the LAC-Z gene has turned non-functional. Functional LAC-Z gene, I will show you, will make blue color. The non-functional will not be able to make the blue color because Z gene enzyme is non-functional. So I will very easily be able to separate the transformants from the non-transformants, and only the transformants will then be grown in a raw material. कॉन्सेप्ट क्लियर? कितने बच्चों को आज पहली बार इंसर्शनल इनएक्टिवेशन और LAC-Z जीन समझ में आया? वी रियली वर्कड हार्ड. अभी तो आप 2 मिनट रुक जाओ, मैं आपको एलाइजा का कॉन्सेप्ट ऐसे क्लियर कराऊंगी, यू विल नेवर फॉरगेट व्हाट इज एंटीजन एंटीबॉडी, ब्लॉटिंग क्या होता है, PCR क्या होता है. कूल? चले आगे. And guys, see, I know you will understand, these are so many technical issues and you know it is lockdown, this is not office, So we are all, especially at Vedantu, we are working so hard for you. हमने लॉकडाउन के बावजूद भी एक दिन का भी छुट्टी नहीं लिया. So even if the voice is a little bit yes or no, up and down, is it fine? Come on, we Biotonians, is that fine? We are all working extremely hard in the lockdown. Yes or no? And where I stay, it's still a red zone. So there is no. We nailed the concept. Yes. Thank you, cutie pie. चलिए, understood? चलो, perfecto, giletto, viletto कर दिया आप लोगों ने. Awesome. चलिए. एक तो ना ये killer slide show है ये.
Plasmids can be good cloning vectors. Molecules will reveal without incorporating foreign DNA. To identify cells that contain plasmids that have incorporated foreign DNA, a second marker gene is needed on the vector. This second marker contains the restriction enzyme site within its nucleotide.
Second one it was.
If foreign DNA is inserted, the second marker is inactivated.
Inactive lac Z.
As insertional inactivation.
It is insertional inactivation.
The marker is the lac Z gene, which codes for the enzyme beta-galactosidase. Beta-galactosidase can cleave a colorless chemical.
Now see beta-galactosidase.
Or a blue compound.
The blue is the non-recombinant.
That harbor the intact vector.
No new DNA, blue.
Can make beta-galactosidase and form a blue color.
Because the lac Z gene will make beta-galactosidase.
However, colonies that contain new recombinant DNA cannot make beta-galactosidase.
No lac Z, no beta-galactosidase. Cream color. Is it clear? So blue color वहीं पर होगा जहां lac Z gene intact थी. Correct everybody. Blue white selection. Blue color where the lac Z gene was intact. White color where the lac Z gene was not intact. Right? So white color for transformants, blue color for non-transformants. This is definitely going to come. It is there in your NCERT. It's a very important concept. It comes in examinations. Crystal clear? Right? हमने यहाँ पर ampicillin resistance का use ही नहीं किया. We were only talking about color change. Correct? Yes? Nailed it? Very good. So now अब आते हैं. अगर मैंने, if I have made a recombinant, have I made a recombinant in the last slide? Did I separate the recombinant with the help of lac Z gene? So I have a plasmid with a foreign gene. I have a recombinant plasmid. मेरे काम का recombinant plasmid. I have been able to manage it. I got a recombinant plasmid which I needed. See? And I have been able to separate it with the help of lac Z gene. Thank you, Snigdha. Understood the concept for the first time, Ruchita? Dinkar? Good. Done, everybody? बिल्कुल दिनकर, बिल्कुल. तो ये रहा. Now will I need millions of copies of it so that I can do biotech? Because मैंने आपको first slide पे I told you it is the scale which matters. I really need a large scale. So will I need hundreds and thousands of copies of it? So can I do something called PCR to amplify the recombinant DNA? Can I do something called PCR to amplify the recombinant DNA? I have got one copy of the recombinant DNA. If I need millions of copies for a large scale of the, see how the steps follow. See how the steps are following. So if I've got a recombinant DNA, can I do polymerase chain reaction to amplify the content of a single recombinant DNA which I've got? And how do I do it? I do it with the help of a technique which is called polymerase chain reaction, which was discovered by a scientist whose name was Carrie Mullis, which comes in the examinations. And now I'm going to teach you about PCR. What we do is we increase the temperature. You know Watson and Crick have told that DNA is semi-conservative in its replication. That means अगर मेरे पास दो strand हैं, मेरे पास दो strand बन गए. Where is the other pen of mine? Oh, it's here only. अगर मैंने ये मेरे पास ही anti-parallel भी बना देती हूँ. देखिए, एक का nib ऊपर, एक का nib नीचे, anti-parallel. If I increase the temperature, will I separate them? और इसके साथ रॉ मटेरियल डाल दूं, इसके साथ रॉ मटेरियल डाल दूं, तो मेरे पास 2 strands बन जाएंगे. ये है एक, फिर से सेपरेट करूंगी, फिर 2 strands. So that is what I have to do. Polymerase chain reaction. ओरिजिनली, वेरी गुड क्वेश्चन, शिखा जी पूछ रहे हैं कि क्या मैम ओरिजिनली इकोलाई के पास कोई रेजिस्टेंस है? नहीं, रेजिस्टेंस सिर्फ प्लास्मिड के पास है, ठीक है। रेजिस्टेंस किसके पास है? प्लास्मिड। क्या इकोलाई के जीन के पास कोई रेजिस्टेंस है? The antibiotic resistance and the resistance is only with the plasmid. Understood? तो हर एक साइकिल के टर्न्स है denaturation, primer and annealing, because आपको पता है डीएनए बनाने के लिए you require a small piece of primer and you will then extend the primer. You will heat it, the strands will separate. I will show you a beautiful video for the same. Correct? Then you will put the primers. You know the new synthesis happens from 5 to 3 direction, and because the temperature is very high, you will go to a bacteria. Again, copycat human being, very smart human being. खुद तो कुछ नहीं किया, इधर से ले, उधर से ले, इधर से ले, उधर से ले और दे दे. Right? That is human tendency. Right? देवियांशु, I remember you. तनहिया, I remember you. So उन्होंने कहा गया वो Thermophilus aquaticus के पास. Thermophilus aquaticus के पास से टैक पॉलीमरेज ले लिया, टैक पॉलीमरेज, और टैक पॉलीमरेज इज थर्मोस्टेबल. ह्यूमन डीएनए पॉलीमरेज इज नॉट थर्मोस्टेबल, सो इट वेंट टू टैक पॉलीमरेज. वहां से ले लिया उसने टैक पॉलीमरेज, Thermophilus aquaticus से एक्सटेंड, and after 30 cycles you will have more than 1 billion copies of the same DNA. Is that clear? Understood? चले? Can we move ahead? Denaturation, annealing, extension. So let's show you another video. Oops, अब चल नहीं रहा है, पहले ऐसे भाग भाग के चल रहा था. चले.
The polymerase chain reaction is a method for making many copies of a specific
strand of DNA.
A very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA.
Even at a criminal place, I can get a very small DNA which I can amplify.
The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase.
These are the raw materials.
The DNA primers to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules.
Heat single strand.
The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the cell.
Primers will anneal. 5 to 3, 5 to 3. Taq polymerase. See the raw materials. See, beautiful video. Raw materials. एक से दो बन गए. फिर से हीट किया, फिर कूल किया, फिर प्राइमर्स, फिर रॉ मटेरियल, टैग पॉलीमरेज.
This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies.
After 21 cycles, over 1 million copies
can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
So we have something which is called a thermocycler, which we normally use. The thermocycler raises and lowers the temperature at appropriate time intervals, discovered by Carey Mueller. This is a process. Taq polymerase is used because it is thermostable. You heat it at 90 degrees Celsius, bring it down to 65 to 72 degrees Celsius. Finish. Done. Any doubts? अब तो सबसे बेस्ट वीडियोस 2 वीडियोस बचे हुए हैं, बट टुडेज क्लास विल बी एक्सटेंडेड बाय हाफ एन आवर. चलेगा? बिकॉज मैराथन है. स्वीटो, यस यस यस, चले. I will do for ecology also. नमीरा, आप टेंशन मत लो. हिमांशु जी, प्लास्मिड बैक्टीरिया में नहीं होता. कई बैक्टीरिया में जिनमें प्लास्मिड होता है उसको F पॉजिटिव कहते हैं. अगर प्लास्मिड ना हो तो उसको F नेगेटिव कहते हैं. नॉट ऑल बैक्टीरिया हैव प्लास्मिड्स. कईयों में ही होता है ना? कईयों में होता है, सब में नहीं होता है. डन? यस, चल आगे बढ़ते हैं. Okay, moving ahead, guys. I hope Humeru, Apurv, Suraj, Aman gang, I remember each one of you. I do remember each one of you. Chaliye, let's move on to the next slide. Now suppose I have made the foreign DNA, मैंने बना लिया, और out of this big human DNA, this big, I just want this much insulin, which is this much. So if I have to tell you in a very simple way what we do, and my aim at Be Biotonic, our aim at Vedantu is always to simplify your knowledge, give you the best possible knowledge, concepts. I always focus on concepts, so that's the core strength. आपके concept strong है, you'll be able to do everything in the simplest possible language. So just see how I'm trying to simplify things for you. But आप लोग will not simplify anything for us. You guys are not going to make sure that you like, share and subscribe to our channel. करोगे? Are you going to make sure that you like, share and subscribe to our channel? बताइए. Yes guys. मैंने कल आपको एक छोटी सी रिक्वेस्ट करी थी और मैंने बोला था बच्चों कि ना आज तक 280 अभी भी देखो सुबह का मैराथन भी हो गया, इतने नॉटी बच्चे तो मैंने कहीं भी नहीं देखे. अच्छा मुझे ये बताओ इससे पहले कि मैं Agarose इलेक्ट्रोफोरेसिस कराऊं, how many of you have told your school friends, if you are in Akendra Vidyalaya, if you are in any school anywhere, have you told your friends about our channel? बताया आप लोगों ने, सच्ची बताइए. राइट 2020 वालों को, 2021 वालों को, अपने जूनियर्स को, 11th में जूनियर्स को, 12th में जूनियर्स को छोटा सा व्हाट्सएप अपडेट किया है आप लोगों ने? पक्का? आर यू टेलिंग? यस, ओके, आई ट्रस्ट यू. यस, छोटा सा इंस्टाग्राम पे एक स्टोरी कर दीजिए, फेसबुक पे एक स्टोरी कर दीजिए, लिटिल थिंग्स. यस, पक्का, प्रॉमिस? चलो. So what we have to do here is that if I have such a huge DNA, such a huge one, my gene of interest is only this much. This is my gene of interest. Can you see my gene of interest, please? Can you see this is my gene of interest? So my GOI, which I'm going to write my gene of interest, is really small in size. So its molecular weight is going to be less. If it's small in size, will its molecular weight be less? Right? So if its molecular weight is less, small in size, will it move faster in Agarose gel versus the bigger one? So I can say that DNA is negatively charged. If the DNA is negatively charged, it will move towards a positively charged anode. And if it is moving towards a positively charged anode, मैं सारे fragments यहाँ डाल रही हूँ. The smallest fragment, let me make that with a black colored pen. So I put all the fragments here. Now the smallest fragments, will obviously be able to move ahead because of low molecular weight. Yes, no, Divya? They will obviously be able to move ahead towards the anode. So this is how, and what I do is I put a dye. What is the name of the dye that I add? Ethidium bromide. And when I watch under ultraviolet radiations, the ethidium bromide, very good Namina. When I watch under the ultraviolet radiations with the help of the ethidium bromide dye, I am able to see orange colored DNA. Which color? Orange color. So let's move ahead and let's understand this. Separation and isolation of the DNA fragments. How do you separate them? You separate them with the help of a restriction endonuclease, right? And then you put them on the Agarose gel to get the separated fragments. DNA आपका negatively charged होता है, anode की तरफ move करता है. DNA fragments को हम size के basis पे separate करते हैं because the smaller the size, the lower will be the molecular weight. You can write that down. Right? Sieving means we can kind of छान सकते हैं. We can separate them with the help of size. ये base का जो polymer है, where they move according to size, is made from sieves. Smaller sized fragments have lower molecular weight and they will move much faster. How will you see them? You will see them with the help of ethidium bromide dye under ultraviolet radiations. चलिए. So now, and what do you finally do? आपके पास plasmid आ गया जिसको आपने कट कर लिया. Gene of interest जो मैं शुरू से insulin gene बोल रही हूँ, you will recombine them with the help of ligase and you will put them inside a host cell. You will give it the raw materials which is needed. अगर ये आपका यहाँ इसने receive कर लिया, इसको 51 amino acids का raw material दे दीजिए because insulin will require A and B chains, 51 amino acids. Got it everybody? And इस 51 amino acid में आप जब raw materials इसको दे देंगे, it will finally be able to make insulin for you. What will it give you? It will finally be able to make insulin for you. Isolation of the genetic material, cutting of the genetic material at specific location. What is the specific location? I am revising it. What is the specific location? Palindrome. What is the specific location? Palindrome. You amplify it using PCR, right? You insert the foreign gene into the host cell, give it raw materials, and downstreaming processes, when you have to sell it in the market, you will find out whether koi allergen to nahi hai, whether it is not causing any allergic reaction, is it safe to use, you will market it, you will test it like how they do for vaccines. So many testing has to be done, animal testing, human testing, and only then will you be able to give it in the market. So this is it, right? Let's look at the entire process.
Cloning a gene involves insertion of a DNA sequence into a piece of autonomously replicating DNA, such as a plasmid. The transfer of this new plasmid construct into a bacterial cell, and the growth of a population of bacterial cells containing the plasmid. In this way, a clone is generated consisting of many exact copies of the inserted sequence. A plasmid contains a bacterial origin of replication that is
plasmid has override
to replicate in a bacterial cell. The plasmid also carries a selectable marker gene that makes the bacterial cell transformed with this plasmid, able to grow in a specific defined medium. In our example here, the plasmid contains a gene that makes the bacteria resistant to the antibiotic ampicillin.
They have antibiotic resistance gene.
Location in the plasmid is a copy of the LAC-Z gene that will produce the enzyme beta-galactosidase when the plasmid is inside the bacterial cell. The LAC-Z gene in the plasmid has a region that is recognized by specific restriction endonuclease.
Palindrome in the LAC-Z gene.
Endonuclease that recognizes this region cuts the plasmid only at this location, so it is a unique site for that enzyme. Most restriction enzymes cut the two DNA strands in different places and thus produce regions of unpaired sequence called
so it will be complementary to that.
The piece of DNA we wish to clone must now be generated. In our example here, we will clone the beta-globin gene obtained from human chromosomal DNA. If we treat human chromosomal
same restriction endonuclease
used to open the cloning vector plasmid, a set of specific fragments will be generated that vary in size. Provided there is not a restriction site inside the beta-globin gene, one of the fragments generated will contain the complete beta-globin gene. However, other fragments of human DNA not containing the beta-globin gene will also be generated. When the fragments of human chromosomal DNA are mixed with the plasmids that
only this will attach.
Restriction endonuclease, the sticky ends of the plasmid DNA
Thank you, Aditya.
Fragments can hybridize with each other, forming double helical structures at the overhangs. These structures can be further stabilized.
Overhangs are sticky ends.
Enzyme DNA ligase. This enzyme ligases DNA strands by covalently connecting breaks that occur in the DNA backbone.
Yes,
This
Namira.
is exactly the situation at the sites where the double-strand overhangs have now hybridized.
Holme, I do recognize you.
Ligase closes
Surya
these
Tapa.
strand breaks, making a new circular DNA cloning vector molecule with a human DNA fragment ligated into its unique restriction site. While some plasmids will receive the human DNA fragment containing the beta-globin gene, other plasmids will be generated containing other fragments of human DNA that do not contain the beta-globin gene. Still, other plasmids will simply have sticky ends that find each other and no human DNA will be inserted. DNA ligase will then close these plasmids again, regenerating
Non-recombinant blue color
reaction. 3 types of cloning vector plasmids will be formed. The population of various plasmids is then transformed into a population of bacterial cells that have been specially treated to make them competent to receive DNA. This generates a population of transformed bacterial cells which contain cloning vector plasmids.
Only this is useful.
DNA inserts. These These cells are then plated onto solid growth media containing ampicillin.
Yes.
These cells that have taken up a plasmid are able to grow and form bacterial colonies. Petri dishes filled with solid growth media plus ampicillin also contain a colorless substance that the LAC-Z gene protein, namely beta-galactosidase, can convert to a blue substance. After overnight growth,
Blue
those
for non-recombinants
from cells that received a plasmid without an insert will appear blue. This is because the LAC-Z gene in these plasmids is able to produce a functional beta-galactosidase. In contrast, colonies that received a plasmid with a human DNA insert will be unable to synthesize beta-galactosidase because the human DNA fragment would disrupt the connection between the LAC-Z gene and its promoter. These colonies will appear white. White colonies can then be selected and examined for the presence of human beta-globin gene. A colony containing this gene can then be saved and grown for further study of the cloned beta-globin gene.
Simple. I will now explain it to you. You saw the video. Whatever I have explained, everything is explained in a gist. I will now explain it to you. Don't worry. होता क्या है? सबसे पहले हमारे पास plasmids होते हैं. Now you ask so many questions. Why ethidium bromide is not used now? Because it is carcinogenic. We have so many antibiotics. Against each antibiotic, we have an antibiotic resistance gene, but they are usually not used. The most common used one is the LAC-Z gene. So होता ये है, if there are 3 cloning vectors, एक के अंदर the LAC-Z will close by it. So this is going to make blue colored colonies because it is the same. एक के अंदर the LAC-Z will open up and the foreign gene will come. The foreign useful gene. This is the one which I need because when I see this is the most important one. हो सकता है other one might have a waste gene which I don't need. Let me make it with blue only. Waste, other gene, useful no, other gene waste. I don't need this. So in दोनों को how will I separate? With the help of white colored. This will be blue, this will be white. So simple it is. Yes or no? How many of you understood? Make hearts. Is that clear? तनिया जी अपना doubt आप main chat section में लिख दीजिए. Insertional inactivation of LAC-Z has happened here. Insertion, and when you see grow this in raw material, you will find it will not make insulin for you, so you will reject this sample because ये आपके लिए gene of interest, insulin gene of interest is only here. So simple, it will not make anything for you. Color कैसे use करते हैं? Blue and white. You will use these colonies. You will throw away these colonies. Because blue color तभी बन रहा है when LAC-Z has not been insertionally inactivated. Right? This is white. That means it is insertional inactivation. That is why LAC-Z gene is not working. And here you will not get any gene of interest product. So you will throw them. So simple if you are understanding. Done? चलिए आगे बढ़ते हैं. अब आ जाओ bioreactors पे. What are bioreactors? You cannot do them in small things. Why can't you do them? अगले 10 minutes में we are moving to the Mahamenti of biotechnology. Mahamenti, super amazing Mahamenti of biotech. And tomorrow we will do NTA अभ्यास tests. Tomorrow सबसे important है कि हमारे concept strong हो. Tomorrow in the morning at 10 o'clock I will do the NTA अभ्यास tests. All the NTA अभ्यास tests join together. Done? आ जाइए. So it is an agitator. What is agitator? You have to control it. You have to move it like this continuously. Why do you want to move it? Why? Because the raw materials need to mix up. That is why I need to move it. The raw material, अगर मैंने insulin के लिए 51 amino acids दिए हैं, that need to move with the plasmid. So I move it. One second, guys. Yes, I need to give oxygen. If there are aerobic bacteria, sometimes too much of temperature causes foaming. There has to be a foam control system, a pH control system, and I contain insulin बन भी रहा है या नहीं? So I take out a sample and see. I again take out a sample and see. So these are all parts of a bioreactor. I have already done it in detail so many times. So you have to give air, you have to keep the thermostat outside to control temperature, sensor probes for pH, for temperature, thermal jacket, and whatever extra material I am taking out, I will see whether everything is getting produced or not. पूजा है ना? चलिए, downstreaming process क्या होता है? देखो बेटा, what is upstreaming when I am making a vector? That is upstreaming. जब मैं vector बना रही हूँ in the laboratory. What is downstreaming? When I need to sell it in the market. It's not easy. When I need to sell it in the market, I need to be very confident about the product. The product cannot cause any allergy. The product cannot cause any reaction. It needs to be a very, very good product. If it causes an allergic reaction, gone. Finish. The company will be closed. Hundreds and thousands of people will die. So I need to purify the products. I cannot give the products just like that. I need to add suitable preservatives. It should not happen that I made the product in Delhi while going to Chennai, it got spoiled. And the vaccine was put into some child and the child did not survive because the vaccine did not have preservatives. The bacteria became active. So the quality, there have to be clinical trials. The quality has to be very good. This process will be known as downstreaming process. You cannot make milk, you cannot make yogurt, you cannot make beta-lactalbumin, you cannot make insulin which is not pure, whose quality control QC checking has not been done. So downstreaming is a process for QC checking, right? चलिए, so genetically engineered insulin बना लेते हैं हम ऐसे. देखो क्या लिखा है, Humulin, it's available. Humulin, human insulin has got 51 amino acids. Banting and Pest were the first people to do it. Eli Lilly Corporation was the first corporation to do it. MCQs. Chain A में 21, chain B में 30. They are joined together by disulfide bonds between two cysteine residues. Cysteine is a sulfur-containing amino acid, right? Along with methionine. तो SS के बीच में disulfide. दोनों chains कैसे जुड़ी होती है? The two chains are joined by SS. Cool. 21 and 30 amino acids. Is that clear? Taniyat understood. Eli Lilly Corporation. Banting and Pest. Gene therapy. Severe combined immunodeficiency disease होती है. कितनी बार MCQs में आया है? कितनी बार? Severe combined immunodeficiency. It is a method to correct a defective gene diagnosed in a child or embryo. Adenosine deaminase deficiency होती है जिसकी वजह से severe combined immunodeficiency. What do we do to overcome ADA? We insert this gene. Is that clear, Janvi? Yes, we are. But yeah, just 10 seconds break from this conversation that we are having. Just another 5. I really never thought that in last 7 or 8 months we will become such a strong, such a cohesive and such a united family. But thank you, my dear children. This is all the power of intention. This is the power of Vedantu, and this is the power of We Biotonic, which I always say. We've become really such a strong family. It's like a bond, the purest bond between a teacher and the students could be seen on this channel. And I've seen hundreds of channels with millions of subscribers, but I'm sometimes so, I see a lot of hate, I see a lot of negativity on those channels. But the moment I come and see this channel, I see India here. I see unity in diversity. We may have difference of opinions. We may have, we may sometimes like or dislike each other, but a true picture of India which Vedantu represents, which was the aim of setting up Vedantu, can be seen, it can be felt. Not only seen, it can be felt at this amazing channel, which is amazing. Yes, no, everybody? Perfect. चलिए आ जाइए. तो बच्चों होता क्या है, जब भी किसी की बॉडी में एडिनोसिन डीअमाइनेज की डेफिशिएंसी होती है, उस पर्सन की बॉडी में इम्यूनोडेफिशिएंसी डिसऑर्डर होता है, जिसको कहते हैं सिवियर कंबाइंड इम्यूनोडेफिशिएंसी. डिफेक्टिव जीन है, देयर इज अ डिफेक्टिव जीन बिकॉज ऑफ विच एडीए नहीं बनता है. दिस वाज द फर्स्ट एवर टेस्ट done for gene therapy in the year 1990 in a 4-year-old girl. ADA deficiency हो जाती है, ADA deficiency, ADA. So what we do is bacteria carrying plasmid with cloned normal ADA gene. Is that clear? And genetically disabled retrovirus, that means ये disease नहीं cause करेगा. It is genetically disabled. No disease. अब इनके T cells में T cells don't have ADA. That is why immunodeficiency is there. T cells के पास ADA नहीं है. Can you see the virus is taking the gene to the T cells? Look at that. Can you see? Hi Holme. Yes, no, Humaira. Malati, can you see the virus carried the gene to the tRNA? See this fast. Can you see? देखो ये ब्लू कलर की जीन वायरस ले गया tRNA के पास. tRNA के पास जीन नहीं थी. Retrovirus, very important. tRNA, the virus carried the gene to the tRNA, and then you made multiple copies. They are implanted in the bone marrow to give ADA. The T cells will make colonies. Can we move on to the MCQs now? चले MCQ करें. Right? BT cotton मैंने आपको लास्ट क्लास में करवा दिया था. I think now we can move on to our MCQs पहले. Menti, start. And what we will do is we will take Biotech 2 for I think ये जो अपना Eliza tested that I can take HHD का एक और मेगा लेक्चर. What say? But आज मेरा Menti because Menti बहुत अच्छा है. Let's check what all knowledge have you gained. Menti, खुशबू, start करें. Menti चलो आ जाओ फिर. BT cotton, BT brinjal तो clear है आप लोगों को, right? BT cotton, BT brinjal is clear to everybody in my class. एक बार RNA interference करवा दूं. RNA interference करा दूं. Menti पहले. Okay. Menti आ जाओ. Menti. Let's start with Menti. आइए. 551482. कोरोना को रोकने के लिए we can't really use RNA interference. I will talk about RNA interference. No worries. पहले आप इस पे फोकस कीजिए. नहीं श्लोका नहीं. We can't do that बच्चे. Maria will do. Five 551482. Menti.com 551482. Submit. I am evil today. My name is Dr. Evil. चलिए. Five 1482. Starting in 5, 4, 3, 2, 1, and I'm starting. Come on, guys. You got a cake. Starting in 5, starting in 4, 3, 2, 1. Start. Snowflake. Chaliye. Nice and tricky questions. I want everybody to do well. Let's see who tops our leaderboard. Awesome class tha. समझ में आया मुक्ता? चलिए, व्हाट इज द एंजाइमेटिक फंक्शन ऑफ रिस्ट्रिक्शन एंजाइम्स? टू ऐड न्यू न्यूक्लियोटाइड्स, टू क्लीव न्यूक्लिक एसिड्स, to join nucleotides during replication or during transcription. Restriction, I told you right in the beginning. Same performance has to be replicated on 26th July. Absolutely same performance has to be replicated on 26th July. होना चाहिए replication. हो जाएगा? Biotonians. Yes, no, Biotonians, exactly same performance needs to be replicated on 26th July. I want replication. Cut, copy, paste. Next question on your screens. है ना? Nice statement of Hossamani. Exactly same performance. चलिए. 5, 4, 3, 2. बताइए. Come on, you guys. Multiple identical copies of a gene for basic research. मेरे तो बायोटेक के स्टूडेंट्स भी कर रहे हैं ये सब. It should ideally have been PCR, but you clone. Clone is a word. Identical copies. Restriction is to cut. बच्चों English language में आपको confuse कर दिया. Ligase is to join. Reverse transcription is DNA to RNA. No, RNA to DNA. DNA to RNA is transcription. RNA to DNA is reverse transcription. Teminism discovered by Temin and Baltimore. Gene cloning. Same performance. Replicate in the exam. Cut, copy, paste. Very good. चलिए. And guys, make sure you are liking, you are supporting our channel, you are loving back Vedantu Biotonic. Almond Gang classes, Bhargavi, I'm going to start from the coming week. I will announce it and start it. You will get notifications. No worries, Bhargavi. Which of the following separates molecules by movement? Easiest question. Come on. Restriction, gene cloning, ligase, electrophoresis. Come on, nail it. Biotonians. Let's stop NEET Biotonians. Excellent. Biotonians are savage people. They will absolutely rip apart the 2020 NEET Bio paper. Yes, no, Biotonians? Very, very cool. Biotonians, आपका जो भी friend है या आप लोग अगर physics में struggle कर रहे हो, तो I can feel you. I literally can feel you. And मैं बता रही हूँ 2 महीने में भी आपकी फिजिक्स स्ट्रगल में बहुत हेल्प हो सकती है. Tomorrow Sunday special, I am taking a physics special. Sunday special, Vani ma'am special, Biotonic special and physics special. पता होना, फिजिक्स स्पेशल लेक्चर ले रही हूँ कल 5 बजे, भूलना मत. सुबह अभ्यास के लिए आ रहे हैं हम लोग. डन. चलिए आ जाइए. Biotonians, completely rock your exam. Own it. Gene therapy आ जाइए. Come on, Biotonians. बताइए. Gene therapy, HIV patients cannot be used. Widely accepted procedure. Bone marrow cells, मैंने एक वर्ड यूज किया था. क्या वर्ड यूज किया था मैंने क्लास में? बताइए. Very good, Biotonians. Very good, Biotonians. Bone marrow, because you give the ADA cells through the retrovirus inside the bone marrow. Very good. Guys, don't forget to show your support. चलिए एक लाइक शेयर सब्सक्राइब हो जाना चाहिए क्लास में. Genetic engineering is used by the pharmaceutical industry. Which one of the following is not? बेटा जी वैक्सीन्स बना रहे हैं हम, प्लांट से, एनिमल से. वी आर मेकिंग वैक्सीन्स. ग्रोथ हार्मोन बना रहे हैं, इंसुलिन बना रहे हैं. क्रिएशन ऑफ प्रोडक्ट्स दैट विल रिमूव पॉइजन फ्रॉम द ह्यूमन बॉडी। अगर ये बन जाए तो क्या मजा ही आ जाए, पॉइजन निकल जाए, ऑल दैट मेंटल पॉइजन भी निकल जाए। यस, आपको पता है कितने आ जाते हैं हमारी क्लास में मेंटल पॉइजन वाले, उनको भी हम यही वैक्सीन लगवा देंगे। एक वैक्सीन हुआ, सारा टॉक्सिसिटी आउट। यस, इतने महीनों से हम काम कर रहे हैं उनकी टॉक्सिसिटी हटाने के लिए, हमने एक पूरी आर्मी तैयार कर दी उनकी टॉक्सिसिटी हटाने के लिए। अगर हमने वैक्सीन बना लिया, वाओ, बट we have not been able to create it. Yes, everybody, I hope you will not forget it now. Yes, Hana, Ishita, we have not been able to create, to remove poisons from the human body. For everything else, there is an answer. For poisonous substances, yet we don't have an answer. Chaliye, next question. चलिए, पीडीएफ अवेलेबल होगा बेटा, क्यों अवेलेबल नहीं होगा, है ना। चलिए, जेनेटिकली engineered plants, BT cotton, BT brinjal, plants. Very good. Cut, copy, paste in your exam. Same performance. Are more difficult than animals? No, it's easier. Are able to fix nitrogen themselves? No. Are banned throughout the world. Include a transgenic rice that can help prevent vitamin A, which is called as golden rice. Right? We require bacteria. We require rhizobium spores. Then only nitrogen fixation will occur. 4-year-old girl from today's class. I will do that, my dear. उमेरू, चलिए, and guys, make sure you like, share, subscribe our channel. We do require your love, we do require your support. चलिए, which kind of therapy was given in 1990? वेरी गुड, इम्यूनोथेरेपी नहीं, उसको जीन थेरेपी कहते हैं। एंटीबॉडीज देने को इम्यूनोथेरेपी कहते हैं। कीमोथेरेपी फॉर कैंसर, बेचारे 4 साल के बच्चे को क्यों मारना चाहते हैं आप, है ना। कीमोथेरेपी नहीं है, इम्यूनोथेरेपी भी नहीं है, नो, इट इज नॉट इम्यूनोथेरेपी। रेडिएशन थेरेपी भी नहीं है, इट वाज गिवन जीन थेरेपी, है ना। चलिए, द इंट्रोडक्शन ऑफ tDNA, ट्रांसजेनिक T. Is it Anis? Concepts are clear, right? I don't think so. Now you will have any problems with your concepts. I want your concepts to be crystal clear. Crystal. tDNA. क्या बात, वेरी गुड। नेक्स्ट वन, इसमें तो एक्सप्लेनेशन की भी जरूरत नहीं है। यस, क्रॉप्स engineered for glyphosate are resistant to, गुड, बताइए ये क्या चीज है। टोटली मैम ने बोला, आई विल डू दिस विद यू। बताइए, पर आप अपनी नॉलेज से बताइए। And guys, today one dedication, एक weekend dedication on your Insta handles, on your DPs, on your WhatsApps to be Biotonic. एक message in your school groups. ये मैंने कराया हुआ है। No बेटा, no. Glyphosate is herbicide resistant. Herbicide, right? Fungus नहीं है, bacteria नहीं है, insect नहीं है, herbicides. Herbicide resistant. Your final leaderboard: Anushka Ghosh and Cherry B, Devanshi Surya, Indrani is a real mascot, Ashwini Divriti, Aparna Singh. बिल्कुल नहीं भूले बच्चे, Earthman Dikhi, वेरी गुड, हैना, राना एंड अंशिका, बिल्कुल नहीं भूलिए पढ़ना, मेरे को रेट्रोग्रेड एमनीसिया नहीं है। आई डू रिमेंबर ईच एंड एवरी वन ऑफ यू। अनुष्का घोष एंड चेरी भी ये साथ मिलके कैसा कंपटीशन खेल रहे हैं। Good guys, amazing performance today. I'm going to see you sharp at 5 o'clock in the evening now. बीच में हो सकता है मैं लाइव आ जाऊं, अगर आप लोगों से कुछ और बात करने का मन करे तो हो सकता है एक छोटा सा लाइव हम कर लें। Right everybody, and in the evening you have 2 bumper sessions today. आज लव सर केमिस्ट्री में बात करने वाले हैं अभ्यास पेपर की, और सूरी सर आपको फिजिक्स सॉल्व करने के कुछ ऑसम कॉन्सेप्ट्स बताने वाले हैं। Tomorrow morning at 10 o'clock we have NT अभ्यास टेस्ट। Bye.